Hi, I'm Steve Palumbi, and this is Lecture 11 in the Science of the Extreme Life of the Sea. Today we're going to talk about how long animals live in the ocean, what their lifespans are, and how we can tell how long they live, because after a couple of decades, it's really hard to keep track of marine critters throughout the whole ocean. So how do we know what their lifespans really are? Where we're going to start is in a pretty strange place, this atoll in the North Pacific. That's Nam Island, but this is Bikini Atoll. And this lagoon piece right here, right next to the island, is not actually a natural part of the lagoon at all. It's a hydrogen bomb crater. This is the Castle Bravo crater. And from 1948 to 1958 or so, the United States did a lot of open air testing of atomic bombs in Bikini Atoll and Eniwetok Atoll nearby. Other nations did similarly, and so as a consequence, those atomic bomb tests in the open air changed the atmosphere. In particular, they threw a number of different kinds of radioactive isotopes into the atmosphere and raised their levels. Now, this is a graph of one of those isotopes. It's called carbon-14. Well, naturally, carbon-14 is fairly low level in the atmosphere. It's made slowly by cosmic rays, but it decays pretty quickly. And as a consequence, we don't really have much of it naturally. But the hydrogen bomb blasts in the early 1960s, in particular, threw a huge amount more C14 into the atmosphere. And you can see it peaked about 1965. Well, that C14 didn't just stay in the atmosphere, it went into the ocean too. At first, it penetrated the ocean pretty shallowly. In the 60s and 70s, it really didn't get deeper than a couple, 100 meters or so. Gradually got deeper and deeper to 500 meters. By then, the C14 count was declining because it has a pretty low half-life. And that signal then of a pulse of C14 right in the 1960s, 70s, and 80s is a kind of a record that we can read in structures that organisms built in that period of time. So this graph, for example, shows one such record from 1910 to 1990. This is the level of C14 in corals in off the coast of Australia and in Fiji. And what you can see is in the 1950s or so, low levels of C14, but those levels increased dramatically into the 1970s. Then they begin to tail off a little bit. So we can see that because corals, after all, are secreting a hard skeleton. And so what they're doing is taking that C14 out of the atmosphere in the ocean, putting it into their skeleton, where we can read that in bands in the coral as the coral has grown during this period of time. Also shown on this graph are the C14 levels of a certain kind of structure that fish make, a kind of a bone. It's their ear bone. It's called an otolith. Otoliths are generally extracted from the ears of fish. They're small calcareous inclusions, much smaller than a rice grain. And if you take them and split them in half and polish them, you can see that they grow in layers. And each of these layers represents usually about a different year in the life of a fish. So the outer layer is the part of the otolith that's laid down most recently, and the core of the otolith is the part that was laid down originally in the fish's larval phase. So the otolith itself has been used as sort of a flight recorder for fish. The conditions the fish was living under when it was small are recorded in the calcium carbonate part of the center of the otolith. And as you move further and further along this set of rings, you're picking up the conditions that the fish was living in at different stages of its life. This is a larval fish. It has an otolith too, that small little core. And the general life history of fish is that they often usually spawn eggs into the water column. Those hatch into a planktonic larval phase, which is shown here. That spends time in the plankton as zooplankton, eating other zooplankton, sometimes the phytoplankton that are there. They metamorphose into juveniles, and that's represented here. Again, the otolith is growing as the fish is growing and is laying down layers day by day that people can count. And then oftentimes, then after the larval fish has metamorphosed, it'll be in its its adult habitat for the rest of its life. But it might have been in a different habitat as a larva. So a lot of deep sea fish have larvae that are shallow, and then they go into the deep sea and stay there. So you're seeing in the otolith the conditions that the fish larva was in, and then later on, as the otolith grows, you're seeing the conditions that the adult was in. So in order to look at the age of fish across this core, you could look at the core, and then you could look at different layers from the core on to the outside of it, and then ask what is the C14 levels in the core and these other layers. And when you do that, what you then see is, well, which fish were making the core of their otolith during the 1950s and 60s and 70s when the C14 levels were high. So by doing that, you can look to see if the core has a high level of C14, or maybe some other ring in the otolith has a high level of C14. Wherever that part is, that tells you that that's when that particular part of the otolith was laid down. That gives you a time check in the otolith, and then from that plus the layers, you can estimate how old the fish actually is. So this represents a graph of the C14 levels of fish otoliths as a function of the birth year of those fish. And you can see that the core of the otolith for fish that are born in the 1940s and 50s have relatively low amounts of C14. As the birth year goes up, the core gets hotter and hotter with C14, and then it begins to decline over time. So by using this kind of data set, you can ask, well, what is the age range of fish all over the world? Because the C14 went all over the world in the atmosphere. And when people did this, they got surprisingly high numbers for fish that otherwise people thought were only living for a decade or so. Groups of fish in tropical Pacific, 8-bar grouper, for example, snappers, are living 30, 40 or more years. Humphead wrasse are living for about 40 years or so. Kelp sea bass, a very big fish, and kelp horse on the west coast, up to 5 decades. And these numbers are not the oldest fish by any means. In fact, a set of deep sea rockfish along the west coast of the United States have been clocked in at living 80, 90, up to 120 years. Well, that set of numbers that set of values can also be applied to other kinds of fish in the ocean. And so we can use the same sort of questions to ask, well, how long do, for example, white sharks live? The trouble here is that white sharks don't have calcareous bones, and so they don't lay down the same kinds of structures that bony fish do. They don't have otoliths, for example, that can be used as a flight recorder. But what they do have is sets of striations in their vertebrae that people had taken as a measure of the years that they had lived. But you have to actually take that kind of assumption and check it with, for example, C14 dating. And when the C14 dating had been applied to white sharks, then yes, it was discovered that those striations in the vertebrae were actually marking the years, but that for some times those striations were missing, or for the larger sharks, some of them were so crunched together that in fact they were missed in the surveys and the counting of those striations. So because of that, the upper ages of white sharks had been underestimated by 20 to 30 years. One study of white sharks in the North Atlantic concluded that the large adult white sharks were living on average 44, 45 to 75 years of age. Same is true of beluga whales, that the kinds of values that we had put on their age were incorrect once we finally calibrated their age with bomb carbon. For example, beluga whales live in very seasonal environments, and so it was thought that in their teeth, which are continuously growing as their as they're aging, they would lay down 2 layers in their teeth each year to reflect the very different seasons that they lived in. But in fact, when those layers were checked with C14, it was found that in fact there was only 1 layer a year. And so an animal that was thought to be 20 years old because it had 40 layers was in fact 40 years old instead. So that changed how we thought the populations of beluga whales could grow over time. A population that, for example, had an average of 30-year-old animals really has an average of 60-year-old animals. Plus, that allowed us to determine that the age at first reproduction, the age of maturity of beluga whales, was actually twice as long as we thought, meaning that it took a lot longer for young growing into the population to get to reproductive age. Probably the prize for this kind of technique goes to estimating the age of Greenland sharks. These animals live in the cold waters of the very northern Atlantic. They get to be very big, are very slow moving, have a very low metabolic rate, and no one had any idea how long they could be living. And in this particular case, the structure that was examined was not an otolith because they don't have them. There weren't striations in their vertebrae because they didn't seem to have those either. But instead, what was looked at was the C14 levels in the eye lenses that these sharks had. Eye lenses are a particularly strange structure because the lens of the eye of a vertebrate, it grows sort of like an otolith. That is, the organism is born with a particular small little lens, and then over time, as the eye grows, the lens gets bigger and bigger by growing layers around it. So that core of the lens is actually a part of the body that was there at the very earliest ages of those animals. And by looking at the core of the eye lens, looking at the radiocarbon in it, then researchers could get an idea of the relative age of these sharks compared to their body length, leading them to have a much better estimate of the actual age of Greenland sharks up to 400 years. This shows the estimated date of birth for a number of different Greenland sharks in this particular study, pushing back here to AD 1600 and the 1700s. That's when some of the old big Greenland sharks alive today were born. In the second part of this lecture on the oldest animals in the ocean, we'll talk about bowhead whales. And we'll talk about how we can try to get an idea of how long they actually live. Bowhead whales, like most mammals, are constantly reconstituting their body parts. And so there's not too many places that you could look in a bowhead whale and find, say, a signature of bomb carbon. Now, in beluga whales, we could because they have the teeth that grow with them, and those are structures that aren't reconfigured over life. But of course, baleen whales don't have teeth, and so how do you actually tell what the age is of an animal as big as, say, a bowhead whale? For a long time, people thought that bowhead whales lived 50, 60, 70 years. That was the average lifespan that most people thought whales had. And then a couple of very odd finds were announced that really changed how people thought about it. And this was one of them. This is a bronze harpoon head. It was found in a scar on the back of a bowhead whale that was hunted by Inuits in northern Canada in the early 1990s. Well, no one has thrown a bronze harpoon at a bowhead whale for over 100 years. So that meant that this harpoon was sitting in that scar on the back of that whale for a very long time. It meant that that whale was very much older than anybody else thought. Subsequent finds, once people started looking, showed that other whales also had harpoons stuck in scars, some of them stone harpoon tips that are, of course, even older than the bronze harpoon shown here. So it really invigorated this quest for trying to find out how long whales really lived and what kinds of structures you could use to ask that question. Among the various mammalian structures that people looked at, there's one that they settled on that actually is laid down early in life and then kept there. And it's the same structure that people use to look at the age of Greenland sharks, and that's the lens of the eye. Now, the lens of the eye is made of protein, and during gestation of a mammal, as the eyes are formed, that lens is formed. And as the animal's born and grows and lives, that lens gets bigger, but the center part of it stays the same. So the protein molecules in that lens are the same molecules over time. They never change. And a whale that's 50 years old still has 50-year-old lens molecules in the center of its eyes. So we knew that the lenses were there. How can you actually say how old those proteins are? Well, a set of chemists, protein chemists, began to look at it and reasoned this way, that amino acids in proteins are all one-handedness. That is, amino acids come in right-handed and left-handed structures, and that has to do with the angle of the carbon bonds. Right-handed amino acids are chemically very similar to left-handed amino acids, but it's only left-handed amino acids that we use in proteins. This just shows the way chemists think about handedness. Left-handed amino acids and right-handed amino acids have the same chemical formula. It's just that the positions of the different groups, the amino group is shown on the right here, and the amino group is shown on the left here. And so that's the handedness of this amino acid. Well, actually, the amino acids can flip from one handedness to another at a certain slow rate. That's called racemization. And these chemists concluded that maybe the rate of racemization would give them the ability to say how old those proteins were. The way they reasoned was that at birth, when the proteins were constructed, they were all left-handed amino acids. But over time, as those proteins were basically inert in the lens of the whale, then racemization would be happening, and some of those left-handed amino acids would naturally and spontaneously convert to right-handed amino acids. So they started measuring the racemization rate of the amino acids in the lenses of whales of different known age to see if their estimates were right. And so what they came up with is essentially a half-life. That is, how long it takes to move half of the amino acids, the left-hand amino acids, to right-handed amino acid. And that varies with the different amino acids. And they measured that with a series of whales of known age and then applied that to a series of whales of unknown age to use this racemization clock to get an idea of the birth date of bowhead whales. This can work not only with whales with all mammals, although the rates of racemization are going to be different from time to time. What these researchers discovered was that for whales of 10 to 12 meters length, the age estimated from racemization was correct. It was in the order of a couple of decades. But by the time you got to animals that were 14, 15, 16 meters long, then the estimate of their age ramped up to well over a century. And what this showed was that as these whales grow, they're adding length as they're aging, but at some point they stop growing. They stay that size, and as they age, of course, they just stay there. But their lenses tell us that they've gotten older and older. It showed that the largest individuals in the group of bowhead whales is made up of individuals that are often over 100 years old. The oldest ones were males, but many of the large females are also over 100 years. Some of them are still pregnant and giving birth, and so it really changed how we thought about the maximum lifespan of these animals. It also showed that it took 30 to 40 years of growth before bowhead whales became sexually mature and started having offspring. And so it told us that the lifespan of these animals was a lot longer, and their life clock, their generation clock, was a lot slower than we had thought before, meaning their populations were not going to grow back as quickly after over harvesting from whaling as we thought in the past. These animals at 100, 120, even almost 200 years old are the oldest mammals known. Greenland sharks are 400 years, small fish off the coast of California are over 100 years. But those are not still the oldest animals that we know of in the ocean. That record goes to this kind of animal. It's a coral growing off the slopes of Hawaii, about 1,000 feet deep. The environment is very, very homogeneous, small currents, no light, not much living around. These animals can get to be 6 to 8 feet high, but they're only growing at about a half a hair's width a year. They're slowly adding size and mass because the deep sea doesn't have a whole lot of food. Well, when these animals were sampled and their ages estimated, then it turns out that this set of species are the oldest animals known on the planet. The record being held by a coral on the slopes of Hawaii that's 4,270 years old. That bespeaks a kind of a lifespan that's about as old as the age of some of the pyramids in Egypt. And it means that the animals themselves are living and growing at a very, very, very slow rate, a very slow clock. They're living this long because they're colonial and they keep growing, and they don't seem to have a maximum lifespan, unlike other animals like bowhead whales and most of the animals we're familiar with. So the next sort of question then is, well, why is it that some animals like these can live for thousands of years, whereas other animals like bowhead whales, even though they grow or they live a long time, they seem to have a maximum age? To answer that, the next lecture is going to be about the theory of aging, what kinds of things happen when animals age, and how evolutionary arguments can help us understand why that is.